Dear friends and my dear students, myself Dr. P.K. Verma. In the first session of Human Physiology, I had discussed anatomy of digestive system and mentioned about the structure of buccal cavity and different parts of alimentary canal. I had also discussed about the anatomy of different parts of alimentary canal. In that session, I had mentioned digestive glands but had not gone into details of their structure and proper function. In this session, therefore, only the digestive glands will be discussed with their details of anatomy and function. You all know that our food contains complex organic compounds, proteins, carbohydrates, and lipids, along with a small amount of nucleic acid, minerals, and vitamin. Because the protein, carbohydrates, and lipids are larger molecules, we need a digestive system by which the food components are digested into simple and soluble substances, absorbed and assimilated in our body. We have got a very efficient digestive system comprising mouth, alimentary canal, digestive glands like salivary glands, gastric glands, intestinal glands, liver and pancreas. At first, we must discuss about the salivary glands. In the human oral cavity, also known as buccal cavity, there are three pairs of salivary glands. These are submandibular, parotid, and sublingual glands. In rabbit, a pair of infraorbital salivary glands also is present. The salivary glands are typical compound tubular glands in which the glandular cells lie within a supporting framework of connective tissue. In this diagram, you can see position of the glands. The uppermost is the parotid gland just in front of the position of our ears, behind the buccal cavity. Its duct is called extension duct that opens in the upper jaw. The second one is just below the tongue. It is sublingual gland and its Bartholin ducts. These are 10 to 20 minute ducts opening in the lower jaw. The third gland is the parotid gland, submandibular gland that opens through Wharton ducts just below the tongue. The glandular cells form a single layer around a larger space known as acinus or alveolus, into which secretion is stored. A number of small intercalated ducts from the adjacent acini join together to form larger duct, and the secretion from the glands finally reach mouth along a larger duct. Such arrangement in which the acini are arranged like a bunch of grapes is known as racemose arrangement. In this diagram, you can see the transverse section. Acinar cells surround a small space enclosing the follicular structure or the duct of salivary gland. These glands are also supplied richly with blood vessels and lymphatic ducts. The sublingual glands respond most quickly to presence of even feeling of a tasteful subject, feeling of the tasteful food, but the submandibular gland secretes highest amount of saliva. These glands open separately by their ducts in the oral cavity when food is chewed. Sublingual gland has the duct known as duct of Ricinus or Bartholin's duct. Parotid gland has the duct named as Stenson's duct, and the submandibular glands has the ducts known as Wharton's duct. The ducts of parotid glands, that is Stenson's duct, enter the mouth opposite the second upper molar teeth. Submandibular glands have a single Wharton's duct which reach the mouth at the side of the frenulum of the tongue. But the secretion of sublingual glands passes by way of some 10 to 20 small Bartholin ducts that open into the floor of the mouth. The parotid glands contain mostly the serous cells, so their secretion is more transparent than other glands and also contains highest amount of enzymes. Sublingual gland is rich in mucus cells, but the mandibular glands has both types of cells roughly in equal number. The salivary glands are supplied with sympathetic and parasympathetic both nerves. In man, about 500 ml saliva is produced per day, but the volume may increase to 1500 ml, in which 70% is contributed by submandibular glands, 25% by parotid gland, and 5% by the sublingual gland. So the maximum contribution of saliva is done by the submandibular glands. In case of fever, saliva is poorly secreted. Salivation decreases also in the conditions of thirst and extreme nervousness. Injection of atropine inhibits salivary secretion. Saliva contains two types of secretions. These are mucus secretions to lubricate the food and the serous secretion in which enzyme alpha amylase, commonly known as ptyalin, and lysozymes are present. Salivary amylase is activated at pH 6.8, that is slightly acidic medium, in presence of chloride ions. The pH is maintained by bicarbonate, phosphate, as well as mucin. In esophagus there is no gland, but in the stomach, especially in the pyloric stomach, there are gastric glands in the pits between gastric villi. These gastric glands contain peptic cells and oxyntic cells. The oxyntic cells are also known as parietal cells. These cells secrete hydrochloric acid. The proton derived from water is released actively by proton pump, and chloride is released passively following proton by symport. The secretion of hydrochloric acid is under direct control of a peptide hormone gastrin released from the wall of pyloric stomach after arrival of food. Human gastrin 1 has molecular weight 2114. It has following composition: glutamic acid, glycine, proline, tryptophan, leucine, glutamic acid, glutamic acid, glutamic acid, glutamic acid, glutamic acid, alanine, tyrosine, glycine, tryptophan, methionine, aspartic acid, and phenylalanine. In gastrin 2, tryptophan is sulfated. The last four amino acids that had been written in bold letters act as active centers of the hormone. Hydrochloric acid is secreted also due to vagus or parasympathetic impulse. The vagus nerve terminals present in the wall of stomach initiate gastric secretion as well as peristalsis. Another cell, peptic cell, is also known as chief cells and zymogen cells. They secrete zymogen granules containing propepsin or pepsinogen. In the acidic medium pH 1.4 to 4.5, the enzyme pepsin is activated and causes hydrolysis of insoluble polypeptides into smaller fragments commonly known as peptones and proteases. Pepsin breaks the peptide bond with tyrosine or phenylalanine. Pepsin does not act on the soluble proteins, therefore cannot digest the soluble milk protein casinogen. In children who are dependent fully on milk, another enzyme renin is secreted that causes precipitation of milk into calcium caseinate. It is commonly called as curdling of milk. Thereafter, the solidified casein can be digested by pepsin. Renin enzyme is not secreted in adults and therefore milk protein is digested very poorly in them. Now come to the intestinal gland. In the intestine, tubular glands are present almost in entire length. In duodenum, there are Brunner's glands that secrete only mucus. But in ileum, especially in the middle part and lower part, there are several crypts of liver cones that secrete intestinal juice or the succus entericus. It contains carboxypeptidase, disaccharides, and a little amount of lipase and nuclease. Now come to a major gland known as liver and the gallbladder associated with it. You can see a large dark red structure that is liver. It is supplied with arteries and veins, and below it there is a greenish gallbladder in this diagram. Liver is the largest gland of our body It is situated inside thorax, just below the level of heart. Towards left side it has 3 lobes, but towards right side it has 2 lobes. Inside each lobe there are a large number of polyhedral liver lobules that looks hexagonal or octagonal in transverse sections. These lobules are separated from each other by a thin layer of fibrous connective tissue forming Gleason's capsule. These are highly vascular and are supplied pure blood from hepatic arteries and deoxygenated but nutrient-rich blood from the hepatic portal vessel. This portal vessel carries blood from intestine to liver, so it is rich in sugar and amino acid. The blood returns through hepatic vein that joins the postcaval vein. The hepatic vein is found in the center of every liver lobule. The hepatic veins lie in the center of each lobule and collect blood from all sides, but the branches of hepatic artery and hepatic portal vessels enter from different corners of the lobules. From the center towards periphery, the hepatic cell cords are present radially and some of them become often branched. These hepatocytes are metabolically very active and they also divide regularly to regenerate any damaged part. In the spaces between cell cords, known as sinusoids, some amoeboid phagocytic cells, known as Kupffer cells, are present. The bile ducts pass outwards along the cell cords and emerge out in the corners near the hepatic artery and portal vessels. Thus, these 3 ducts are known as hepatic triad. Hepatic triad thus include hepatic artery, hepatic portal vein, and the bile duct. Now, this is the transverse section of liver. In some of the lobules, you can see an empty part from which the hepatic vein originates. In between the lobules, you can see the fibrous connective tissue. At some places it is thinner, but at some places it is wider. This is the arrangement of one particular lobule. Here in this diagram, 6 corners have been shown, and in each corner, the red color represents hepatic artery, blue color represents hepatic portal vein, and green color represents bile duct. So these 3 ducts together form the portal triad or hepatic triad. See an enlarged view of the arrangement. You can see the cell cords originating from the center towards outside. In between 2 cell cords, there are fine bile ducts that terminate in a major bile duct in the corner. In the middle, portal vein, and in another side, the portal artery is shown. It is diagrammatic representation of a cell cord around bile duct. A few amoeboid Kupffer cells also have been shown in the diagram. Inside liver, the following functions are performed. The liver stores glucose in the form of glycogen. This function is known as glycogenesis. In case of hunger, the glycogen breaks into glucose and supplied to blood. Breakdown of glycogen is known as glycogenolysis. Glycogenesis is under control of hormone insulin, whereas glycogenolysis is under control of glucagon. Both these hormones are secreted by islets of Langerhans in the pancreas. About 90% of plasma proteins are synthesized in liver. That includes the fibrinogen, prothrombin, transferrin, as well as angiotensinogen. In liver, amino acids as well as fatty acids are oxidized. The ammonia released from amino acids convert into urea. Therefore, hepatic veins have highest concentration of urea. The vein returning from liver to the posterior vein is known as hepatic vein. It has highest concentration of urea. Liver converts carotene into vitamin A, retinol and retinaldehyde, and these are stored for as many as 6 months. Liver oxidizes alcohol into aldehyde and can derive energy for metabolism from it. Therefore, in alcoholic patients, extra fat deposits in liver, causing liver cirrhosis. The old and dead RBC is destroyed in liver. Its heme molecule breaks into bilirubin that, when comes into blood, forms biliverdin. Liver is the largest site of steroid biosynthesis. Cholesterol formed in it converts into bile salts, the glycocolate and taurocolate used in digestion of fat. It is the center of detoxification where many drugs and toxins can be destroyed. In this way, liver is a very useful organ in our body. Now, the metabolic products in liver pass through the bile duct and become stored in the gallbladder. It contains bile salts, bile pigments, along with many other organic and inorganic materials. These are released into duodenum for excretion, but many of them may be useful in the process of digestion also. The gallbladder is an unpaired structure. It has a thin wall containing only a thin layer of muscles and columnar epithelium. The epithelial cells absorb water and inorganic salts, so the bile stored in gallbladder becomes highly concentrated. Bile becomes more viscous due to mixing of mucus. In human, hardly 32 milliliter bile can be stored in gallbladder, as it is revealed from X-ray photograph and the ultrasonography. The cystic duct joint forming common hepatic duct that opens in duodenum. The pancreatic duct also opens near the ampulla of bile duct known as ampulla of Vater. At their opening, there is the valve of Oddi, originally described by Gleason in 1654. Bile contains 89% water and 11% solid, including bile salt 6%, cholesterol 2.5%, phospholipids 0.4%, inorganic salt 0.8%, and the pH ranges between 7 and 7.6. When chyme comes into duodenum, bile mixes with it. Due to presence of high concentration of bicarbonates in the bile, chyme is neutralized and its pH is raised to 7.6, at which the pancreatic enzymes can act well. See the structure of pancreas. It has a head part, the middle is body, and the terminal part is its tail. Its duct, that is pancreatic duct, opens jointly with the bile duct in duodenum. Pancreas is a compound gland that has about 60 to 62% part made of enzyme secreting exocrine cells known as acinar cells. The acinar cells form small follicles at the tip of fine pancreatic ducts. In the connective tissue, only 6 to 7% part is made of endocrine cells, forming the bunch called islets of Langerhans. Acinar cells release proenzymes of different digestive enzymes into the follicle and carried away by the ducts. All such ducts join together and open into duodenum by a common pancreatic duct near the ampulla of common bile duct. The pancreatic enzymes are released under control of duodenal peptide hormone secretin as well as pancreogymin. The pancreatic juice contains alpha amylase, lipase, trypsinogen, chymotrypsinogen, and carboxypeptidase, along with a small amount of nuclease and elastase. The islets of Langerhans comprise as many as 3 types of cells. The alpha cells produce glucagon, beta cells produce insulin, and delta cell produce gastrin and somatostatin. This is the photograph of the transverse section of pancreas. You can see connective tissue in between the acinar cells. When the connective tissue is magnified, at some places you can see the bunch of interstitial cells forming islet of Langerhans. This is the photograph of the islet of Langerhans. So pancreas is endocrine as well as exocrine part. It is a composite gland or compound gland. In the next session, we will discuss the physiology of digestion and absorption. See you very soon. Thank you. Thank you very much.